cross-pollination,
This call is now being recorded.
autogamy, geitonogamy, and xenogamy. We discussed all the agents of cross-pollination, biotic and biotic agents. Now we'll talk about the outbreeding devices, that means contrivances. That means for the adaptations for cross-pollination. That means what is the reason that here in these case, these plants, only cross-pollination is possible and self-pollination doesn't take place, can't possible, is not possible or doesn't take place. First thing, now first reason one by one we will see. So these are considered to be outbreeding devices, that means contrivances. That means only cross pollination is possible and self pollination is not possible. Now what are the reasons? One by one we'll see. First is unisexuality. Uni means single, means that flower, that plant contains, that flower is not bisexual, that means it contains only one sex organ, either male or female. So in that case, uh, only cross pollination is possible. If plant is bisexual, that flower is bisexual, then only there are chances of self pollination. So if the plant is having only one of the sex organs, either male or female, then cross pollination only is possible. So self pollination is not possible in this case. Plants may be monoecious, for example, maize, or dioecious like mulberry or papaya. Monoecious means what? The plant is the single plant. Mono is single. That same plant is having both the male and female or reproductive organs, but flowers are different. Dioecious means two different plants. Male plant is different, female plant is different. Means dioecious, monoecious nature. That day was I was telling you while explaining you hydrophily, isn't it? That monoecious nature is what? Within the same plant body, both male and female reproductive plants are parts are present. Maybe as a male flower and a female flower. But dioecious nature is what? The two plants are different, means male plant is totally different, female plant is totally different. So in dioecious, dio is two. So in the both the cases, then the cross pollination only is possible. Okay, when it is unisexuality is the case. Now second is dicogamy. Dico means again di is related to two. Means what? Anthers and stigmas, which are considered to be male and female reproductive parts of the flower. They mature at different times in bisexual flower. Means the flower is bisexual, but the maturity of these sex organs, that is the male and female, is different. So that self-pollination is not possible. So that is the reason that dicogamy, where only cross-pollination is possible. Now there are two types of dicogamy. When male reproductive organ, that means anthers, mature first than the female, that is called as protandry. Protandry is what? It is related to androecium. Andry is related to androecium. Pro is first. That means what? Androecium matures first, earlier than the gynoecium. So example of this is what? Disc florets of sunflower. Now that you know that there are two types of florets in case of sunflower. So these disc florets of sunflower, their anthers mature earlier than gynoecium. Okay, and protogyny means what? Pro is again earlier, first. Gyne is what? Gynoecium. So gynoecium matures earlier than androecium. That is the another case where that you can understand that protogyny and protandry, these are the two different types of dicogamy. That means the maturation period of the male and female sex organs is different. In protandry, anthers, that means the male reproductive part matures earlier than female, and in protogyny, female, that means gynoecium, matures earlier than male reproductive part, that means androecium. So this is the another way where only cross pollination is possible. Now, prepotency. Now here also, see, pre is earlier, potential, that means what? Pollen grains of other flowers germinate rapidly over the stigma than the pollen grains from the same flower. Means that in this case, the flower is bisexual. That means male and female both carpel and the androecium and gynoecium are present. The maturity period might also be same, similar. But here, when the pollen grains of the other flower are fallen on the stigma of that particular plant, these pollen grains they fertilize, germinate rapidly than the pollen grains of the same flower. So pre-potency, please earlier. First, potentiality of other pollen grains, that means pollen grains of the other flowers, it is more than its own pollen grains. So this is the third criteria where only cross pollination is possible. Then second, next is hercogamy. Again, here is another criteria where only cross pollination is possible. Here is the mechanical device to prevent self pollination in a bisexual flower. Mechanically, that in that plant, naturally, plant itself is a barrier. That means those floral parts, that means the sex organ itself, themselves, they become physical barrier. That means what? Their height or the position of these sex organs is made naturally in such a way that they cannot come in contact with each other easily, so that it avoids the contact of pollens with the stigma of the same flower. Example, calotropis, where pentagular stigma is positioned above the level of anthers. That means what? Stigma is at the top and level of the anthers is below it, so that how the pollen grains which are produced in the anthers of the same flower will be reached to the stigma of the same flower if it is, its position is made naturally in such a way that it itself becomes the barrier, physical barrier, so that the pollen grains of the same flower cannot reach the stigma of the same flower. So this is what the mechanical device which prevents self pollination in bisexual flowers. Okay. Now second, next fifth criteria where only cross pollination is possible is that self incompatibility, means self sterility. Sterile means what? Not able to fertile. That means in such flowers, though the pollen grains of the same flower, though the flower is bisexual, though the maturation period is same, though the position is physically naturally normal, but when the pollen grains of the same flower fall on the stigma of the same flower, then these pollen grains cannot germinate, but they become fertile. They remain, sorry, they remain sterile. They cannot fertile the stigma, that means the ovule, that means the stigma cannot accept. Though the flower or the pollen grains fall on the stigma, it cannot be germinated. That means it is called as self incompatibility. That is not that is also called as self sterility. That means the same pollen grain cannot germinate and is not able to fertile the ovule, that particular female reproductive organ within the same flower. So this is called as self sterility or self incompatibility. Now this is the picture where in this it is shown the entire pollination types, where in this case you can see just a minute, entire structure of the model of pollination pictures shown. First, self pollination divided into autogamy. Autogamy again of two types, chasmogamy and crystogamy, and then cross pollination. Then self pollination is again considered to be one as autogamy, the other as geitonogamy. Then cross pollination is between the two different plants. So this is just in short, it's shown. Now we will be seeing what are the advantages of self-pollination, what are the disadvantages of the self-pollination. First thing, in case of self-pollination, first advantage we can consider is that there is no dependency on the pollinating agents. That means pollinating agents are not required. So it saves a lot of energy with this. And Second advantage you can consider is that the parental characters or race purity is maintained. That means if you want to maintain the pure form, that particular character in the same next progeny also for several generations, then that particular character can be maintained. That is called as purity. Purity of that particular character is maintained with the help of only self-pollination. Then second is what? Recessive characters are eliminated in this self pollination. And the second is next is seed production is ensured with less number of pollen production. That means, as I said, that seed production is ensured because it is self pollination, there are no pollinating agents, and so it is surety. So surety, purity, that is the main important thing in case of self pollination. Surety in the sense sure method of pollination because both the sex organs are present, bisexual flower. Then purity of the gametes, means gametes, that means whatever the characters are maintained, that purity of that particular character is maintained. Third is that recessive characters can be eliminated, and the next is what? Production is ensured. That means it is sure method where seeds can be produced by this way, and that also with less number of pollen production, because as there is no loss of pollen grains, there is no requirement of agents. Obviously, there is no loss. That energy is saved, and there is no loss of pollen grains. Now, as this at the same time, we have to consider or this study or focus on the disadvantages of the self pollination also. So what are these? First and the most important thing in case of self pollination which can be considered as disadvantage is new and useful characters are not introduced, like disease resistance capacity or environmental adaptability. So such new characters or vigor, even some cases vigor also, so such important and useful new characters cannot be introduced if it is pure form. That means it is the first disadvantage which you should remember always by the self pollination. Then second important thing is that because of continuous self pollination for few generations, like 4 to 5 generations, or even after that also, that there is decrease in the vigor and vitality of the race due to inbreeding depression. What happens? Continuously, the same plant is continued for self pollination for several generations, like 4 or 5 generations. Then there are chances of decreasing the vigor, the production is decreased, the vitality is also decreased, and so it affects the overall inbreeding method of self pollination. Then the next we will talk about the cross pollination advantages. Now a few points are there which are advantages of self pollination can be considered as the disadvantages of cross pollination, and advantages of cross pollination can be considered as the disadvantages of self pollination. So it's very simple and easy to remember also. Now, first advantage is that genetic combination introduces variations leading to improved adaptability to environmental changes. When you can observe that when the plants are self-pollinated, they cannot resist to the changes in the environmental conditions like drought, heavy rain, or very high temperature, etc. Whatever the environmental changes are there, that self-pollinated plant which generally cannot withstand with this with such changes. But if you observe that these there is a combination of genetic breeds, right? So genetic variation is obviously there, but these variations, that those combinations which are introducing these important things like improved adaptability to the environmental changes, though there is a drought, very heavy rain, or very high temperature, so such type of environmental changes are adapted by cross-pollinated plants. And increased disease resistance. So obviously, as two different parents are combined, so two different genetic characters are involved in that particular plant by this method, there is disease resistance also increased. And second is increased seed production. Seed production is also increased. vigor is increased, and new varieties are formed. Obviously, as genetically two different plants are there, obviously the combination of these two will give rise to new genetic characters. So new characters are developed. But one thing, that few disadvantages of cross pollinations that we need to consider is first, most important, as dependency upon pollinating agents. As we have discussed, that there are pollinating agents required, abiotic or biotic, whether water, wind as abiotic, and birds, insects, bats, snakes, etc. Biotic agents. So dependency is there for cross pollination. This is the first and the most important point in disadvantages of cross pollination. And as there is dependency, the number of pollen grains produced should be more. So most of the times, especially in case of wind pollinated plants, there is vestige of pollen grains. So loss of energy is there. So lot of energy is wasted during that process. Then large amount of pollen grains are produced, as I told, there is wastage of pollen grains. And then one more thing which is considered in this case is factor of chance is always involved. That means it's not the sure method of pollination. We can't say that surely it will get pollinated by this agent or that agent. There are always chances. Maybe 100% chances of cross pollination are there, but 10% are not there. So it's not sure method of pollination. Cross pollination cannot be considered as a sure method of pollination, because there might happen that pollination may happen or may not happen. So that is why considered to be, it's a factor of chance always. And some undesirable characters may develop and some good characters may get eliminated. Unlike self pollination, here undesirable characters also may get developed, and some good characters also might be eliminated. So this is also one more chance. These are also the probabilities, you can say. It may also happen that undesirable characters are introduced and desirable characters are eliminated. So this is just a chance. Just like it's not the sure method, this is also the chance of this important thing. Now we'll talk about the next part in this, that is the pollen pistil interaction. Now, once you know that which is the male reproductive part, which is the female reproductive part in the plant, which is the reproductive organ, which are the vegetative organs, then from there we have studied the structure of male and female reproductive parts, its development also we have studied. Now, the methods also we have studied through which the further process of fertilization takes place, that is the pollination, the agents, types, everything we have discussed. Now we have come to reach to this point. Then once the pollen grains fall on the stigma, that is the female reproductive organ, stigma, style, and ovary. These are the three parts of the pistil or carpel. Carpel is the female reproductive organ, that is gynecium. A single is called as carpel or pistil. So when the topmost part of this gynecium is called as stigma, right? So pollen grains when fall on the stigma, then what happens? There is the middle way. Middle way is what? Pollination. That transfer of pollen grains from anthers to the stigma. That means from male to the female reproductive, as both are non-motile. That's why this pollination is important. Now, once pollination is done, completed, that means pollens fall on the pistil, then what is the further process that we have reached up to this? Okay, it begins with the pollination and ends with fertilization. Remember, once now pollen grains fall on the stigma, then what happens? This is the end of pollination and beginning of the next process, that is fertilization. Okay, pollination is just the middle way, pathway for reaching the male reproductive part to the female reproductive part. Now what happens? Now what are the further events taking place that we are going to discuss right now? All the events from deposition of pollen grain on the stigma to the entry of pollen tube in the ovule are referred as pollen pistil interactions. Now, how the interaction of these pollen grains with the stigma or the pistil, that means carpel, that explains that we will study now. Pollination doesn't guarantee the transfer of right type of pollen. Just now I was talking because wind doesn't know where to fall. Take the pollen grain. Even insects don't know. Means these are all chances. So this is not a sure method. It doesn't give that the transfer of these pollen grains will fall on the right stigma. So these are all just chances of cross pollination. But still has this ability to recognize and accept the right and compatible pollen of the same species. So this is what, though pollen grain of some another species falls on the stigma of the another species, that particular stigma will not receive the pollen grain of the another species. That means what, this pistil is having the ability to recognize and accept the right and compatible pollen of the same species. Thus, wrong type of pollen is discarded by the pistil. It will not get germinate on the stigma. Okay, so compatibility and incompatibility of the pollen pistil is determined by spatial proteins, and this process involves pollen recognition followed by promotion or inhibition of pollen. That means what, if the pollen grain of the same species is fallen on the stigma of the same species, then only that stigma is having the capacity to recognize the pollen grain of the same species, and then only that stigma will release the proteins, special type of proteins, and then only that those pollen grains will develop the pollen tube. So it will promote those sugary substances secreted by the stigma will be helpful for the pollen grain of the same species to promote the pollen tube. And for another species, the pollen grains of the another species, if they are there pollen on the stigma, then it will inhibit the pollen tube formation. Okay, so the compatible pollen absorb water and nutrients from the surface of stigma and then germinate and produces pollen tube. This is what this part is also very important, that still is having this capability to recognize the pollen grains of the same species. Now, once the pollen grains of the same species are recognized, then the further process will start. That means which we learned in the previous part of development of male reproductive organ, where male gametophyte is developed. So the pollen tube is finally that which is formed there. It is finally pushed through the ovule and reaches the embryo sac. Now, what is that connected part, that tubular structure from stigma? It through the style, it will reach to the ovule, correct? Ovule or ovules, wherever, whichever. The ovule reaches the ovule which contains the, reaches the ovule which is present in the ovary. Ovary, style, and stigma. These are the parts of carpel or pistil, or you can say gynoecium. So once the pollen tube is formed, it will start growing, and it grows through the style and reaches the ovule. That is the embryo sac in the ovule. The tip of the pollen tube enters in one of the synergids and then ruptures to release the contents. And due to pistil interaction, intense competition develops even in the compatible pollen grains, means gametes. So it plays very important role in the sexual reproduction and seed formation. Now, what is that? I will explain you with the help of diagrams. Okay, but before that, just I will show you the diagram, and then the another part I will just come there. Okay, before that, just let me tell you this artificial method of hybridization also. This is what we discussed, is what natural way of pollination and the further process of tube formation, pollen pistil interaction. But one more method is also there, that is called as artificial hybridization. This is what self or cross pollination natural way we discussed, but there is artificial way of hybridization where it's one of the major approaches used in the crop improvement nowadays. Only the desired pollen grains are hand pollinated and used for fertilization, and this is accomplished through emasculation and bagging procedure. Okay, so emasculation means what? E masculine. Just try to understand. Split this word. E removal. Masculine, that is what male. Masculine nature means what? Male nature. What is the male part in case of flower? That is the anther, right? So removal of male part from the bisexual flower female plant to avoid self-pollination, that is what emasculation. Simple way you remember, E was is removal, masculation, masculine part, that means male part is removed. And what is the male part in the flower? That is the anther. So anthers are removed from bisexual female parent to avoid self-pollination. This part I will explain you in detail later also in another chapter, but right now you just try to understand as this is a part of pollination, because this is the natural way which we discussed earlier just now, yesterday and today also. But this is the artificial way. Artificially also, we can select by the method of, in the method of hybridization, we can select the male and female parents. And if the female parent is having bisexual flower, then in that case you need to remove the male sexual part. That means anthers need to be removed. If we are using that plant as a female parent, then in that case removal of anthers is must. And that removal of anthers from the bisexual female parent is called as emasculation, to avoid self pollination. And at the same time, bagging is also one more method, because though the anthers from the same flower are removed, there are chances of falling down of unwanted pollen grains in the female flower, though the flower is emasculated. So to avoid it, the emasculated flowers are covered by a bag to avoid unwanted process of cross pollination. So that is called as bagging. And tagging is also one more point that we will discuss later. So this is, just remember, that is the another way in the sense what? This is the artificial way of hybridization. Naturally pollination is taking place, but artificially also by the process of hybridization, we select the male and female parents. And when the female parent is bisexual, in that case that male part is to be removed, and that through emasculation. In detail, we will discuss that part later. Now, after that, after the emasculation and all that artificial, whether it is artificial or a natural way of pollination, once there is recognition of the pollen grains and there is a formation of pollen tube which grows through the style of the pistil or carpel and reaches the ovule in the ovary, then what happens? The next part is called as fertilization. And in that also there is competition. There are many pollen grains of the same species. So many pollen tubes are trying to reach to the ovule. So out of that, then from among them also, the most what you can say, capable with the highest capacity will reach the stigma, will reach to the embryo sac of the ovule. And that there is also the competition between the male gametes, or the gametes which are developed in the pollen tube. You know that the second mitotic division takes place of the generative nucleus to form two male gametes, right? So now this is the next step now, that is fertilization. Now this fertilization in case of angiosperms is called as double fertilization. You know why double? Because, as if you remember, that from the pollen tube, these two male gametes which are produced by the mitotic division of the generative nucleus, right? Generative cell, both the male gametes are utilized and two times the fertilization is taking place, and that's why this is called as double fertilization, which is the unique feature or characteristic feature of angiosperms, because both the male gametes are involved and utilized for the fertilization. That means at two places twice this fertilization takes place, and that's why it is called as double fertilization. Now how it takes place, that I'll explain you. Now, there are some terminologies you need to remember. Siphonogamy means what? Siphon means tube. What is the tube here? Pollen tube, which is carrying two non-motile male gametes, because these male gametes are non-motile. That's why this is the agency of siphon. That means the pollen tube. Siphon means tube. Which is the tube here? That is the pollen tube. So pollen tube is helping this non-motile male gamete to reach to the female gamete, right? So these non-motile male gametes are carried to the ovule through a tube, and this process is called as siphonogamy. As in, when I tell you something different, meaning of that particular word, write it down in the textbook itself. Now, second terminology is what? Porogamy. Pore means small opening, right? So entry of this pollen grains to the ovule is through micropyle. If you remember the structure of angiospermic ovule, the inverted ovule, you can observe that small opening is there present, that integuments are not fused. There is small opening, and that opening is called as micropyle. So the entry of this pollen tube is what? Through micropyle, and that is what called as porogamy. So through that pollen tube, these pollen grains, they enter through the small opening that is called as micropyle, and that is called as porogamy. If this pollen tube enters through chaliza, then it is called as chalizogamy. Exactly opposite to that micropyle, that end is called as chaliza. If that pollen tube enters through chalizal end, that is called as chalizogamy. And if this pollen tube, which is carrying pollen grains of these male gametes, it is entering through the integuments. There are two integuments, outer and inner, So through the walls, then it is called as mesogamy. So entry of pollen tube, actually it's not pollen grain, it's pollen tube which is carrying male gamete. Okay, so if the pollen tube is entering through the ovule, entering the ovule through the integument, then it is called as mesogamy. So these are the three things, ways of entry of pollen tube. One is through micropyle, called as porogamy. The second is through chaliza, called as chalizogamy. The third way is through integuments. It is called as mesogamy. Okay. So these are the three ways of entry of pollen tube, which is carrying the pollen, male gametes, that means pollen grains, obviously male gametes, to reach the female gamete. Now this is the structure. Now I can show you with the help of this diagram, where you can see this anatropous ovule, where this is the, here is the stigma, this is the pollen grain, this is the formation of pollen tube, and this is through the style, this is the style of gynoecium, that means the carpel. This pollen tube is reaching now through this small opening called as micropyle. Now here in this picture, this pollen tube reaches the ovule, and it enters through the micropyle, right? This is the micropyle. This is the micropylar end. Okay, so this is what called as porogamy. And as these pollen grains, that means the male gametes are carried by the pollen tube, this is called as siphonogamy. Now what happens? Once this pollen tube reaches the female gametophyte, this is the embryo sac. Here, this is the egg apparatus. I have already explained this to the micropylar end. Three cells are present. The centralmost cell is called as egg, and the two neighboring cells, sister cells, are called as synergids. This pollen tube first reaches, when it enters through one of the synergids out of these two, because it is having the filiform structures. I have shown finger-like projections, which favors the entry of the pollen tube. Okay, and these finger-like projections or these structures, they help the male gametes to reach the female gamete. So this one of the synergids, through one of the synergids, these pollen tube enters, and then that tube, that means pollen tube gets burst, it gets degenerated, and the two male gametes are released over there. Now once the male gametes are released over there, one of the male gamete fuses with the centralmost cell of the egg apparatus, that is the egg cell, that means the female gamete, and the another one fuses with the secondary nucleus. So at two places, the first, when it fuses with the egg cell, one of the male gametes when fuses with the egg cell, that is called as first fertilization, and that is also called as syngamy. And when the second male gamete, which is released here by the bursting of the pollen tube, that reaches the secondary nucleus, and here the second fertilization takes place, that is called as second fertilization, and this is also called as triple fusion, because secondary nucleus is already diploid, and then when the second male gamete fuses with it, that becomes triploid in nature. Diploid plus one more haploid, that becomes triploid in nature. Okay, so this is what I will show you again with the help of another diagram. Just try to understand with this first. So this is what I told you. Syngamy means what? Syngamy is the first fertilization. That means fusion of one of the male gametes with the egg cell. Egg cell is nothing but called as female gamete to produce zygote. So one male gamete and the one female gamete, both are haploid. So the result of these two haploid gametes is what? Zygote, which is first diploid cell in the process of fertilization. And this fusion of male gamete with the female gamete, that means egg cell, is called as syngamy or first fertilization, and the result of first fertilization is the production of zygote, which is the first diploid cell in the process of fertilization. And the second male gamete, which is carried by the pollen tube, that fuses with the secondary nucleus, that is also called as definitive nucleus, that is already diploid in nature, that results in the formation of triploid primary endosperm nucleus, and that is called as triple fusion or second fertilization. I hope you have understood this. So syngamy is the type of generative fertilization, whereas triple fusion is a type of vegetative fertilization. What is that generative and vegetative? I'll explain you it later. Just try to understand this. I will explain remaining part with the help of another slides. Okay, so that it will be our revision also. Hmm? Everything again I will revise so that we can um, see few more important things with this outbreeding devices. Starting with the outbreeding devices, I will revise what are these outbreeding devices. Dichogamy, as I told you, that is the flowering plants have developed many of the adaptations or contrivances for to discourage self-pollination and encourage cross-pollination. So in some species, pollen release and stigma receptivity are not synchronized. That means what? Maturity period is different. Dichogamy, maturity period is different. That means male and female sex organs, they mature at different time. So protandry in case of sunflower, anthers, that means protandry. Pro is previous, androecium, that means male part. Reproduce, they show maturity earlier than the female part. Whereas in case of datura and solanum, what happens? Gynoecium matures earlier than the androecium. Female parts matures earlier. Trichogamy, that is what? The position. Mechanically, it is naturally it becomes a barrier. Now, in case of hibiscus, very easily you can observe that the stigma is at the topmost position, and so the anthers produced, that means pollen grains are produced on the lower level. So there are no chances of reaching the pollen grains of the same flower to the stigma of the same flower. So hibiscus and gloriosa, these are the examples where the position of these gametes itself becomes barrier. Okay, then the next is heterostyle. Heterostyle, that means the style, that means the either the stigma, that means the style of the female part, that means gynoecium is too high so that anthers cannot reach in the same plant, or the anthers, that means this what you can see, the filament of the anthers, means in androecium, which is made up of anthers, and the stamen is made up of anthers and filament. Filament is having such a long length that also it avoids the self pollination, cannot reach on the stigma of the same flower. So that is what the third another way to prevent the self pollination. Heights of the styles of the flower of the same species is not same, it's different. That's fine. Then the next example is what? Self incompatibility. So where the it prevents the inbreeding of the self pollen grains of the same species of the same flower cannot fertile. They are incapable of fertility. They are not fertile, but they are sterile. But of the another species, another flower, if they are fall, then they are sterile. So only cross pollination is possible. Diclamy, that is the genetic mechanism, prevents self-pollination from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil, which is related to pistil pollen-pistil interaction. It will decline the further development of the pollen grains on the what is self incompatibility also you can say. So it will decline, or what happens as we discuss self incompatibility, pollen grains will not be fertilized in the same flower. See, so this is also another device which favors only cross pollination, avoids, discourages self pollination. So these are some of the examples where several species of papaya, male and female flowers are present on different plants. And so in such case, that is dioecious nature, only there is chance of cross pollination and not the self pollination. Now, pollen pistil interaction here, as we discussed, that pistil is having that capability to recognize the pollen grain. If pollen grains of another species they are fallen on the stigma, it will deny the further process of fertilization. It will no inhibit the further development of those pollen grains, but it will promote the development of the pollen grains of the same species. So this is what called as pollen pistil interaction. Okay. Now, fertilization, the different ways we discussed, that is the entry of pollen tube into the ovule. Porogamy, that means when the pollen tube is entering through the pore, that means micropyle, that is called as porogamy. When the pollen tube is entering through the chaliza end, chaliza, that is called as chalizogamy. When the pollen tube is entering through the integuments, then it is called as mesogamy. So these are the three ways of entry of pollen tube. Now, entry of pollen tube into the embryo sac. Now, irrespective of the place of entry of pollen tube into the ovule, the tube invariably enters the embryo sac at micropylar end, that is degenerating synergid cells, as I told you just now. Means whichever the way the pollen tube gets entry, whether it is through micropyle, whether it is through chaliza, or whether it is entering through the integuments, it doesn't matter. But the way of entry of this pollen tube, once it is entered in this embryo sac, then through the that micropylar end, that means through the one of the synergids only, that pollen tube will enter. That means these are through entry of the pollen tube. Once it is there through the one of the synergids, then only the further process takes place. Now here you can, I will show you the video tomorrow because it takes time. But here see, this is the micropyle carrying the vegetative nucleus and the two male gametes. Once this pollen tube reaches here, you can see here, this is the secondary nucleus, this is the egg cell, this is one of the synergids, right? So this is the synergids and this is the filiform apparatus, that means having finger like projections. So through which this pollen tube enters. Okay. And once it enters, it gets burst. See, you can see here, once this pollen tube enters through this one of the synergids or sister cells, here pollen tube is burst. It degenerates. And once it gets degenerated, the vegetative nucleus also gets degenerated, which is at the tip of the pollen tube, and these two male gametes are released free, right? So one of the male gametes, it joins or it combines it with the male gamete, and there is the first fertilization. Out of these two male gametes, one will fertile or it will combine with the this egg cell, that is the female gamete, and this is called as syngamy or first fertilization. And the result of first fertilization is what? Zygote, formation of zygote, that is the first diploid cell in the fertilization. Now the second male gamete, which will be released out of the synergids and will reach the secondary male gamete, a secondary nucleus, which is already diploid in structure. So when the second male gamete fuses with the secondary nucleus, it forms the triploid nature. So that result of this fusion, that is the second fertilization, because second male gamete is utilized and fertilized. So this is called as second fertilization, and the result of second fertilization is the formation of triploid form. What is that triploid? And this is also called as triploid fusion, because already it is second diploid nature of secondary nucleus. When it fuses with the second male gamete, then it becomes triploid in nature. So this is called as triple fusion, and the form which is produced is called as triploid primary endosperm nucleus. And here it is called as generative. Just note the last sentence when I told you that I will explain it later. It is first fertilization is considered to be generative type, because male gamete and female gametes are fused here. Because that means what? That is related to generative way. That means male gametic. There is a fusion of gametes. So that is called as generative way. And here is the vegetative way, because though second male gamete is utilized, this is the secondary nucleus produced from the vegetative part, that cell. So this is what called as a further process as vegetative way, and this is the generative way of fertilization. Okay, so only this much you remember, nothing else. This is more important. So, and this is what about I told you about the artificial way, where nowadays to improve the crop plant, so many varieties have been developed by using this artificial way of hybridization. And this is what you can see, the anthers are removed with the help of forceps or by hand. There are different ways of removing the anthers. See, with the help of forceps, these anthers are removed from the female part, and that is called as emasculation. And this is what called as bagging. These emasculated flowers, though the anthers male part is removed from the female part, there are chances of falling off anthers of the another species. That means unwanted pollen grains can fall on the same emasculated flower. So to avoid it, these are covered by bags. This is called as bagging. Okay, and so once the stigma becomes receptive, then the bags are removed, and then interested pollen grains which we have selected as the male part, then these are dropped. This dusting of the selected pollen grains is done, and then again these flowers are bagged. That means covered. That is called as the rebagging. Okay, so this is what double fertilization, where you need to remember all these terminologies as syngamy. So the first fertilization is called as syngamy, and the result of first fertilization is the diploid cell called as zygote. Whereas the second fertilization is called as triple fusion, and the result of triple fusion is primary endosperm nucleus. Okay, so this is again shown here, the release of this one sperm cells, that means the pollen tubes. Here is the entry, this is the zygote formation, and this is the endosperm which is produced. That means second fertilization gives rise to endosperm formation, and the first fertilization result is zygote formation. So this is what you we have learned in this chapter. This is again short summary about the events seen in the fertilization process, and this is what you can consider or you have to remember as the very unique feature of the angiospermic plants, that is the double fertilization. Okay, so now if you have any doubts, you can ask me.